to using them as mixtures has seen the contribution of quite a few microbiologists from around the world. From a humble understanding and usage of microbes for curd, cheese, and wine, etc., they have reached a stage where human health seems to be dependent on their contribution on our metabolic pathways. From the 16th century onwards, many theories were framed as to which microbes flourished and spread. The microbiology in India has recorded a history of around 8 decades, and the science of microbiology is still developing rapidly. The Microbiologist Society India was founded around 1996 among the large number of stalwarts who took up the challenge to take Indian contributions in microbiology to a global level. And yet again, with such a discussion, I, on behalf of B.N. Patel Institute of Paramedical and Science, Paramedical Division, managed by Sardar Patel Education Trust, gives me immense pleasure and privilege to welcome you all on 7th interactive session on pioneers in microbiology in India. Speaking of the trust, the Sardar Patel Education Trust has a team of enthusiastic, young, and dedicated and innovative teachers. The institution running under the trust continue to grow, and they have carved a niche for themselves within the span of their existence. The trust's mission is to equip young boys and girls with versatile development of their personalities and making young boys and girls strong morally, emotionally, spiritually, and physically. Talking about our institute, B.N. Patel Institute of Paramedical and Science was established in year 2010 for reinventing healthcare education and to render career opportunities in allied health. Our institute provides various medical and academic facilities. B.N. Patel Institute provides the finest education in paramedical health science in India, and hereby we help students to spread their wings, theoretical as well as practical, in respective fields of specialization offered by our institute. Now, moving further, before we start this session, I feel honored and obliged on behalf of B.N. Patel Institute to invite our eminent speaker, Dr. Swaranjit Singh, Director of SAF Polyclinic, Perch, Punjab. We welcome you, sir. I'm also beholden and grateful to welcome the Chief Patron for today's session, Dr. A.M. Deshmukh, sir, President of Microbiology Society, India, and Shri B.N. Patel, sir, the Managing Trustee and Secretary of Sardar Patel Education Trust. We welcome you, sir. I'm also enlightened to welcome the patrons for this session, Dr. Sanjeev Patankar, President of MBSI Maharashtra and Goa region, Dr. Arpita Arorah, ma'am, Director of B.N. Patel Institute of Paramedical and Science, and the coordinator for today's session, Dr. D.K. Acharya, sir, Associate Professor of B.N. Patel Institute of Paramedical and Science. I welcome you all. To begin with this session, I would like to once again invite Dr. Swaranjit Singh, Director of SAS Free Polyclinic, Perch, Punjab. And to add in his lines, Dr. Swaranjit Singh has contributed in the field of environmental biotechnology, ecology, bio-nanotechnology, and biosurfactant production. Dr. Swaranjit Singh provides expert advice on environmental problems, bioremediations, solid and liquid waste management, and removal of pollutants from various companies. His publications are in high impact factors, journals, and the articles are highly cited in hydrocarbon pollution, effects on living organisms, remediation of contaminated environments, and effects of heavy metal, co-contamination, and biocontamination in USA. To add, he was also a member of NBA Chennai and 2 WFCC task force. One of his excellence has served his excellent administrative management, license skills, and RDA experience as a senior principal scientist and head. Dr. Swaranjit Singh is also an advisor at HP and Punjab government for setting up colleges and universities. In the 70th Foundation Day of CSIR on 26 September, 2 of his very publications were in the list of 70 best publications of CSI based on high Cited Index. Dr. Swaranjit Singh was inducted as a member of Bhopal Action Plan in and around UCIL last year from CSR. He's also an advisor at SD College of Pharmacy, Surat, in from 2016. Sir has also served many lectures of colleges and schools and has also interacted with them to popularize science topics. He's also a fellow of Association of Microbiologists. He's also a fellow member at Society of Applied Biotechnology. He's also once again a fellow member of National Academy of Biological Sciences. Sir is also a fellow member at National Environmental Science Academy. He has been awarded the Award for Excellence in Environmental Management, again, one of the environmental award. Sir has also been awarded the first prize for essay on Team CSI in Labs and Field by DG CSIR. He is once again the member of National Academic of Science India. Sir has also got first prize in bilingual competition on My Ideas on Disaster Management, Kobe University in Japan. A very well awarded sir. Sir has also got as Loyola Award in Chennai in 2011. To add one more line for Sir, Dr. Swaranjit Singh has over 142 publications in high impact journals and has over 32 book chapters. He has also edited a book on nanobiomedicine in Stadium Press in Houston in USA. He is also an editor and reviewer in many of the international journals. Sir, today on behalf of Dean Patel, we are really proud and honored to have you with us. Thank you so much for being with us today. Thank you, sir.
Thank you.
Thank you. Over to you, sir.
Thank you very much for the nice introduction you gave me. I'm so happy to see all the audience and liked your introduction very much.
Thank you, sir.
So we'll go over to the first speaker now. We'll go over to the first pioneer.
Yes, sir. Sure.
Okay. The first pioneer is Ponduri Venkata Ramana Rao. Yes. So the first pioneer in microbiology is Dr. Ponduri Venkata Ramana Rao, who was born on 3rd April 1917 and he lived till 13 April 2005. He is an Indian microbiologist. He was born at Rajupalam in Ongole district, Madras, during the British India. He graduated with a Bachelor's of Science degree from the Government Arts College, Rajamundry, in 1937 and graduated with a Bachelor's of Medicine Surgery degree from Andhra Medical College, Visakhapatnam, in the year 1944. He was in the medical service between 1944 to 1947 and worked as regimental medical officer, field ambulance officer, Army in the Second World War. He obtained a diploma in public health from the All India Institute of Hygiene and Public Health, Kolkata. This is a very old institute in India. In 1948, an MD degree in bacteriology from Andhra Medical College. He joined the Osmania Medical College, Hyderabad, as a lecturer in bacteriology in the year 1953 and was promoted as professor in the year 1957. During his 14 years of service in medical college, he organized the Department of Microbiology and developed into a full-fledged postgraduate center with comprehensive research facilities. He traveled on a ATCM fellowship in 1958 for 1 year to Syracuse, New York, and Virus Lab in Albany, New York, USA, where he worked as yaws, endemic typhus, and cholera. He served as a member of 12 national and international scientific associations and was elected as Fellow American Public Health Association for his work in the field of public health. He was on several ICMR committees and scientific advisory boards. He took over as director as early as 1967 of the Institute of Preventive Medicine, where he worked as the director till 1973. He was instrumental in developing 50 acres of land at Nacharam into premium vaccine unit. Just imagine, in the 1960s, he started vaccine unit. This ambitious project was inaugurated by the President of India, Mr. V. Giri. He visited various institutes in Yugoslavia, United Kingdoms, USSR as a WHO fellow to observe the production of lyophilized smallpox vaccine. What a wonderful research he has done. After retirement, he taught in various medical colleges in Belgium, Devangray, Deccan, and Bidar Dental College. He consulted for the Government of India and the WHO on the National Smallpox Eradication Program, NSEP, in South Bihar. Smallpox was ultimately eradicated from India in the year 1975. This is all I have to tell you about him. We'll have the questions in the last, at the end of the session.
Sure, sir, sure.
Okay.
Definitely, sir.
So we go to the second pioneer in microbiology, that is Edgar J. DaSilva. Smart guy. Edgar J. DaSilva was born on 21st August 1941 and he lived till 28 October 2007. He is a great Indian microbiologist whose passion for the practical applications of his subject had driven him to champion biotechnology in developing countries around the world. DaSilva joined the United Nations Educational, Scientific, and Cultural Organizations. This is known in short as UNESCO in 1974. And he rose to be the head of Life Sciences section at his foresight and perception of science in the global context allowed him to be proactive in identifying scientific priorities and developing new and appropriate thrust for UNESCO's program action. He was instrumental in the planning and implementation of several UNESCO region and international programs in applied microbiology and biotechnology, as well in the development of global networks dealing with the management and use of microbial resources and training opportunities in the field of marine and plant biotechnology. So he has got a varied interest, a very diverse interest of science. He was a co-founder of the Microbiological Resource Center. This is actually a culture collection which houses a lot of cultures. It is known as MIRSAN all around the world and played a major role in the establishment of the Biotechnology Action Council, BAC. Though these two programs, he made a considerable contribution to the development of biotechnology, research, and training worldwide. During his time with UNESCO, DaSilva had a lot of passion for teaching and knowledge transfer in his own area of specialization of microbiology and also in biotechnology. He has got a long contribution to the world. DaSilva's knack for developing strong links with international programs for coordinated action on topics of common interest led to his strong support for the International Organization of Biotechnology and Bioengineering, IOBB, built strong links with the World Federation of Culture Collection. Yes, this is WFCC. And all culture collections have to be members of WFCC. Played an important role in the conference on the global impact of applied microbiology, GIAM, and many others. In addition, he mobilized several extra budget programs in close cooperation with UNEP and UNDP and donor member states for activities in national development in biotechnology and regional cooperation in microbiology. He worked very hard to develop training initiatives and set up fellowship and professorship programs to stimulate scientific exchange. He was a strong proponent of international scientific collaboration as a means to promote development and provide developing countries with the tools to drive their own scientific research. This is all about him. So now we have a lady pioneer, which Dr. Acharya gave me yesterday. A lady pioneer. I'm just coming to that. Her name is Professor Dr. Rameshwari Thakur. You can see her photograph on the right side. She's a former professor who is MBBS, MD in microbiology. She is from Muzaffarnagar Medical College, UP, India. She was born in 1951, 2nd August, in Ahmednagar, Maharashtra, India. Her fields of interest are mycology and dermatophytes and cryptococcus. First to report meningitis due to C. gatti in Botswana. Various publications in reputed international journals indexed in PubMed. By the way, she's a great lady, over 70 years of age, but still she is very active. She graduated MBBS and did postgraduate MD from Indira Gandhi Medical College, Shimla, HP, in the year 1993. Recently, she has been honored with the prestigious international award, ISSN category, International Remarkable Researcher Award in the field of microbiology, rank Golden Research Award 2021. She was first to report the outbreak of Trichophyton indotinea from India and has many publications on this field. She has published around 50 articles in reputed national and international journals. She has more than 850 citations. She is also editor of some reputed national and international journals and participated in various national and international conferences. She worked in research teaching institutes in India and abroad. Being a very dedicated teacher, she takes keen interest in teaching both undergraduate and postgraduate students. She was actively involved in research in Botswana in collaboration with the University of Pennsylvania during HIV/AIDS pandemic from 1999 to 2010. Her field of interest are antibiotic resistant, cryptococcal, and other opportunist infections in patients with HIV, AIDS, and dermatophytosis. In Botswana, she worked as special microbiologist, infection control consultant, institutional review board coordinator, and senior research microbiologist at National Health Laboratory and Princess Marina Hospital, Gaborone. She attended various workshops, conferences, and trainings in South Africa and United States. She was first to report Cryptococcus due to Cryptococcus gati from Botswana. This is all about Madam. Now we have one more 1 year in service director of CFTRI, Mysore. She is the present director of CFTRI Food Laboratory in Mysore. I met her several times in my career as a scientist. She is an accomplished food technologist, an active researcher. She has taken over as director on January 21. After passing from CFTRI with MSc Food Technology in 1988, she worked with Kisan Products Limited, Bangalore, for a short stint before returning to Alma Mater in 1991. She grew up to the position of chief scientist and head of departments of protein chemistry and technology before taking over as the director. She received her doctoral degree in food science under the supervision of Dr. A. G. Apurao. During 96 to 98, she has been with the Institute in Microbiology, Hanover, Germany, on a DARD fellowship. In her career spanning over 3 decades, she worked extensively on both basic and applied aspects of food science. Significant contributions were made towards protein unfolding, structure-function activity relationship, protein and enzyme as food ingredients, nutraceuticals from traditional foods, and technologies for combating malnutrition. Some of these technologies that are transferred to industry include enzyme amylase, rich energy food, soy protein hydrolysate, heat-resistant sesame seeds, and supplementary food for severely malnourished children, SAM, and so on. Over 40 entrepreneurs have taken these technologies so far. During 12th five-year plan period as principal investigator of the Wellness Food Project, an array of innovative products were brought out targeted to every sex of population. Further, a pilot-level malnutrition intervention studies were undertaken during the same period with the participation of over 200 Anganwadi children in order to assess the efficiency of supplementary foods developed by CFTRI. These supplementary foods actually are nutraceuticals also, which have a lot of nutrients in them. She has published over 50 manuscripts in peer-reviewed journals, and she holds a total of 11 patents to her credit, including 5 US patents. Dr. Sridevi has guided 5 students for doctoral thesis, along with other 40 PG students so far. Currently, she is focusing on alternate and unconventional sources of proteins and micronutrients to combat malnutrition and development of novel food integrants for health benefits. She is a bright lady, a gold medalist in graduation from University of Mysore. Sridevi has represented as a young fellow scientist, Hanover, Germany, INF Craft Fellow 2006, Indo-Finland Exchange Program 2018, Speaker in Knowledge Summit at Lyon, France 2019, etc. Dr. Sridevi has been the governing council member of National Agri Biotechnology Institute, Mohali, that is near Chandigarh in Punjab. She is a member expert committee on public health and nutrition, BIS, subcommittee on oils and oil seeds, interagency on micronutrients, ICMR, and board of studies of the Mysore University. She has been the life member of many professional bodies, such as the Association of Food Scientists and Technologists, India, Society of Biological Chemists, India, National Society, Nutritional Society of India, AMI, Association of Microbiologists of India, etc., etc. That is all I have to say today about the pioneers in microbiology. Thank you.
Thank you, sir. Thank you. Thank you so much. Indeed, it was great. Thank you for sharing your knowledge with us today. To all the participants who are there with us today, any question from your side, we are free to answer.
There is no question which is silly.
Any question?
There is no question which is silly.
Yes, sir.
You can ask any question away from the subject also, on microbes, on nutrition, on nutraceuticals, anything you can ask.
Sir, I have one question.
Yes, sir.
Sir, recently one microorganism discovered, which is claimed it is one of the longest size of microorganism. So,
Yes.
what do you think about this discovery?
Yeah, I recently gave a speech on the microbiome day in Kashmir University, Srinagar, on the 27th, and I mentioned about this microbe. It is 1 centimeter long. It is filamentous. It is found in the Caribbean islands, in the Caribbean Sea, a place known as Aruba, in the mangrove area, Aruba. You know, 16S rRNA sequencing is the best method for bacteria. It complies with that. The microbe complies with 16S rRNA, and it is a bacteria. I'm not remembering the name right now. I'll tell you later on. So it's a new discovery. So the word microbe in this case is not pertinent. I'm sure Indian science will do something and the international science will do something, and will not call it microorganism anymore. It may call it a big microorganism, something like that, but it is actually matching with a microbe sequenced. I also was really surprised, 1 centimeter is very big. It is something like mushroom. Mushroom also is a microorganism. It's a fungi. So even mushroom we can see very well, but still it is a microbe. So if you compare with mushroom, it is nothing astonishing. It is normal. But many people don't know that mushroom is a microbe. So I'm sure they will reclassify microbe and big microbe, something like that they will do. Or some new technique may come, which will say by they have different things inside it later on. Yes.
Thank you, sir.
Thank
it is
you,
very
sir.
fascinating.
Any
very
other
fascinating. Yeah. I want to tell you something. I want to tell you something in connection with microbiome. See, microbiome is a collection of microbes. It is not one microbe. It is trillions of microbes on the skin, trillions of microbes in the water, in the pond. If you remove some of the microbes from the microbiome, it does not function. It becomes functionless. That means each set of microbes are dependent on the other set of microbes to do a function. So you cannot, break up the microbiome. It has to be together. This message I want to give all of you, that microbiome and the microbes should be conserved, should be preserved for our benefit.
Thank you, sir. Thank you. So we have got a small suggestion in the chat box from Dr. S.R. Dave. He says that there are few of the microbiologists. There are more, and some other. We will talk about them. So he has given us this, a message to you, from Dr. S.R. Dave.
Can you repeat what he's saying?
Dr. S.R. Dave says that these are the few of the microbiologists. There are more. Some other will talk about them.
Sure.
Small,
sir, I want to say that whatever you explain is very nice, because in one day talk we cannot include all those who are above 70 pioneering microbiologists. There are few more pioneering microbiologists, those who are in contact with them. No, so this series of the talk may continue, and some other people may include some other also.
Let me tell you one thing. This is the 7th session I'm giving today. I have already covered 24 pioneers in microbiology till now.
Okay.
And today we have covered 4 of them. So this makes 28. I have covered 28 pioneers in microbiology today. And yes, as you say, we'll try to cover many more this year.
Okay. I was not fortunate to attend all. Sometime the link was not available to me, and sometime the time was not continuous. So if I will know all the list of the 28, then I would be very happy for this microbiology.
What is your good name?
I'm S.R. Dave.
Okay, all right.
Okay, because
sir, we are going to publish a book on all these microbiologists who are pioneers, and with their snap and CV. And it will be available to you very soon.
Definitely, because as you say, this is the 7th session, so I thought only this 4 or 5 you have talked about. I was not having the other. I have not attended the other one.
Very soon, with your good wishes, we plan to take out a booklet of all these pioneers in microbiology. And in one of the sessions, I covered only women microbiologists. That was a great session about a month back. And I covered 4 women.
So
Means
I'm sure that
Indian women are very.
I'm sure that you must have covered even the JV part.
Not yet.
Sir, good morning, sir. Dave sir, good morning, sir. Dhaval Acharya here, sir. Sir, your suggestion is already accepted because we have already this name. Bhatt sir name is there in our list. So in next May session we will include.
Okay. Means because
we made a one list of pioneer microbiologists, and among that Bhatt sir is one of them.
Okay, means you have made the list and accordingly you are including the. Okay.
Sir, one whole committee is there about this session, and we are screening the pioneering workers who work in the field of the microbiology and the biotechnology, and on the basis of that, the Swaranjit Singh sir, Deshmukh sir is going to decide.
Very good. I
and
agree,
then,
because
yes,
no, we cannot cover all in a
yes, sir, yes,
two
sir.
session. So
Yes.
I thought this is the only session.
No,
I was
no.
not having the
Okay.
much detail about this.
We are organizing such kind of session every month, sir.
Okay.
Every week.
Sorry, every week. Yes.
Okay.
One thing more, I want to request all the participants, if you know some good pioneer in microbiologist who is 70 and above, let us know. We will try to cover that microbiology. It's my appeal to all of you.
Definitely, sir. Definitely. Thank you once again, sir. So we have got one more question for you in the chat section. This question is from Pornan, a PhD student, Presidency College, Chennai, Tamil Nadu. He's thanking you for the great session once again. And his question is, he would like to know about the current status of GMOs and their testing outside of lab. What are the effects that can be expected? Will nature allow such modifications? Also, how stable will be gene therapy considering the natural trend to revert back?
Yeah. GMOs are good, but their release to environment is very restricted. We cannot, release the GMO to the soil without permission, because it may lead to havoc. So very few GMOs are in the soil now. And the second part of the question is reverting back. In the soil, there are trillions and trillions of microbes. They'll communicate with each other, different microbiomes, killer strains. So there'll be interaction between them, and they're liable to change with time. We cannot keep them stable. There'll be genetic change over the years. That's why they say that we have to conserve microbial diversity of today. Let me give you a small example. Penicillium, which produces penicillin. After 50 years or 100 years, it may change genetically. So our children, our youngsters will not have access to penicillin, which is a wonder drug. So penicillium fungi is conserved in many, many culture collections of the world, so that we have the present day penicillium with us. It should not change with time. So supposing penicillium loses the capacity to produce penicillin, the culture collections will give the strain to the society, take and produce penicillin once again. That's why take-home message today is conserve microbial diversity, conserve microbiome. Thank you.
Thank you, sir. Thank you. Any more questions from the participant side? You're welcome to bring it on.
Thank you very much, sir, very much for your wonderful talk. I just wanted to ask another small question. There was a recent message from our group saying that there's a very long, about 30 years or more of experiment to study the different changes of that occur in E. coli. So that long-standing experiment, what do you think the outcomes would be? Will there be any microbes that are totally changing their genetic structure, or will there be evolution continuously putting inputs to make them sturdier to survive this harsh environment of anthropogenic activities, or will there be any extinctions in microbial species? Is there anything like that noted so far, sir? Thank you very much. I'm Purnan here from Chennai.
WFCC, they have a committee, endangered species, endangered microbial species. I'm a member of that committee. Many microbes are vanishing with time. They are unable to bear the harsh environmental conditions. So we have recommended, like I just told you, we should preserve these in culture collections around the world. Every country has got so many culture collections. The government is spending billions and trillions of dollars for maintaining this culture collection. So to avoid the extinction of these microbes, we have to preserve them properly for long term, because genetic change is an ongoing process. It will never stop. Even the humans are changing. There are some birds like tragopan which are finishing. The tortoise, many tortoise in the turtles in the ocean, they are vanishing because lots of pollutants are going to the sea, to the ocean. So, and a lot of plastic is also there, which they are eating, the turtles and the tortoise, and they are dying because of suffocation. So first thing, environment pollution is related to the extinction. So we have to control the environment. We have to control and have a clean environment so that the plants, humans, insects, and the microbes thrive and do not go away from this planet.
Thank you very much. Yeah. Thank you very much, sir. Thank you very much.
Thank you. Thank you very much, sir. Thank you. So any more questions before we end this session?
1 minute, please. Hello.
Sure,
yes,
sir.
hello.
Sure. Yes, sir. You're audible, sir.
Yeah. Hello.
Yes, sir.
Can you hear me?
Yes.
Yes, sir. You're audible quite nicely.
Yes. Thank you. Thank you very much.
Audible, sir. Welcome.
Today I am very happy that the eminent professor in microbiology, Dr. Swaranjit Singh, a really devoted and a dedicated person in microbiology, keeping charge students of microbiology in India by giving knowledge on every Sunday. And today's lecture is also a recorded lecture. We are going to circulate in all groups. And whatsoever the knowledge he has given regarding the biodiversity, really, it was also wonderful knowledge. And it has helped to encourage, to inspire teachers or researchers in microbiology and biotechnology from India. Now, I will reiterate that Dr. Swaranjit Singh is my good friend and the good person also. And with the help of such personalities in India, I'm sure we'll do miracle in biotechnology and microbiology in India. I'm also thankful to Dr. Vivek Upasani. He has attended today, Nilima ma'am from Baramati. And DK Acharya sir also arranging the event taking efforts to arrange such events. Tambuli sir from Umarga, the Udgir also has joined. Thanks everyone. Thank you again.
Thank you.
Thank you so much. Thank you.
Now, Dr. Acharya can give the word of thanks.
Sure, sir. Sure. Thank you. Thank you, sir. Thank you so much for being there with us today. It was very inspiring. See, thank you, sir.
Thank you.
Upasni sir, you raised a hand. So if you have any question, you can please. Upasni sir. Vivek Upasni sir. Hello, Upasni sir. Okay. Thank you very much for giving me the opportunity, Priyal madam, for giving the vote of thanks over here. Here I would like to.
Can you hear me?
Yes,
Yes.
yes, sir.
Hello?
Yes, sir. Yes, yes, sir. Yes, sir.
Yes, sir.
Okay, very good morning to all of you. Good morning, Deshmukh sir, Dave sir, Swaranjit Singh sir, and all the participants. It was a very interesting session. I have been attending on and off, and it's nice to know about the various the development of microbiology in this country. Along with that, I have a suggestion with the food technology. Can we make a database of the technologies available and with whom and how they can be available, from whom it is available? That will give more impetus to the microbiologists and the entrepreneurs in students to develop as entrepreneurs when they know about the technology, where it is available, what is the cost, and how it could be implemented. I think that is also an important aspect, along with just knowing the scientists and just knowing that they have developed certain technologies. So I think that can be done as part of an MSI activity.
Yeah, let me give one comment on this. CFTRI is the leading food technology laboratory in the country. So you can get in connection if you want food technology with CFTRI, and they will tell you all the technologies which are available with them. They will not hide anything. They'll give you all the technologies which you can buy from them or work in them. And if you want if you want bioremediation, the laboratory in Maharashtra, NIRI, is the best one. You have to approach NIRI, Nagpur, and they will tell you all about bioremediation and pollution control and things like that. So you have to write to CSIR headquarters or to the laboratory directly, addressed to the director of that laboratory, and you'll get all the information.
True, sir. True. That is very correct. But many people don't go to the site. So we can have a session where the director of such institutes or some leading scientist from that institute speaks about those technologies, and that would be a good idea, I think.
We have invited speech by many directors. This time we'll try to get some CSI director and tell about his technologies. For example, if we want petroleum technology, we have to go to Institute of Petroleum, Indian Institute of Petroleum, Dehradun. So we can arrange 3-4 directors to talk about the technologies they have produced. Sure, this is a good idea.
Thank you very much, sir. Thank you very much.
Thank you very much, sir, for giving me an opportunity to organize this webinar. Here I would like to thank, first of all, on the behalf of B.N. Patel Institute of Paramedical and the Science, to the speaker, Swaranjit Singh sir, who got contacted by Deshmukh sir, and then he approached me for arranging this all webinar. And since last 2 or 3 days, we communicate lots of things with Swaranjit Singh sir, and I also would like to thank for giving me the knowledge regarding to these things also. Thank you very much, sir. I would also like to thank Deshmukh sir for he is actually giving the lot of different things to the students, and among that this is one of the important activity, and from on the basis of this we can write down the whatever our goal is, that is one book regarding to the pioneer microbiologist of India. So this is one of the good idea. So thank you very much, Deshmukh sir. I would also like to extend my thanks to our college management and the trustee and the secretary, Dr. B.N. Patel sir, as well as the director, Madam Dr. Arpita Arorah, Madam, who gave the permission for the organizing the such kind of subjective events for us. So I would like to thank also to them. I also would like to extend my vote of thanks to the participants as well as the Priyal madam for hosting these all the events very nicely. Thank you very much, who are also hearing on the YouTube also. Thank you very much. Thank you very much, everyone.
Thank you very
Thank
much.
you. Thank you so much, sir. As we have come to the end of the session, once again, I thank everyone for being present with us today. Thank you for sharing your ideas with us. Thank you for here. And thank you, everybody, all the participants. Thank you for making this session a great success, and have a great day ahead.
We will meet next week again. We'll meet next
Thank
week.
you. Thank you.
Sure, sir.
Thank
Sure.
you all.
Indeed, very sure, sir.
Deshmukh Saab.
Yes, yes. Very nice. Thank you. Thank you. Thank you.
All right. See you next time.
Deshmukh sir.